All right, if there's no questions, let's begin. Can you see my screen?
Yes.
Thank you. All right, so what we're doing today is Lab 01, Ubiquity of Microorganisms. You should also find and read the Infectious Disease Research Project. It's a file on the Canvas webpage. And read the project instructions. I'm going to talk about them next week, so that's why I want you to read about them. Also remember that you have the online plagiarism quiz. You need to take the tutorial and take the quiz by 11:59 p.m. this Saturday, April 10th. You must achieve 100% on the plagiarism quiz, or else you'll get 0 for the infectious disease research project, and that is a 100-point project, so you don't want a 0. You may take the plagiarism quiz something like 5 to 7 times. Let me turn on a light here, and then let me remind you that I am going to be at a jury duty Tuesday, April 20th. And so on Tuesday, April 20th, we will have the lab, but I'm going to record it earlier. And the lab will be at 5:30, although, like I said, it'll be recorded. And then at 5 o'clock. And then at 6:30, instead of having the lab, we'll have the lecture on that day, because I think I will be home by 6:30. Any questions about any of that? All right. So that's what we're doing today. If there's no questions, let's close this and talk about Lab 1. And I should point out to you that Canvas has been acting up. This term, only one thing has happened. When I rolled over the class from last term, Lab Module 0 did not roll over, and I don't know why that wasn't the case, because most of the rest of the class is here. So if there is a problem with Canvas, please let me know, and I will do my best to correct it. Last term there were lots of problems with Canvas. So far I've only had one problem, and that one was sort of strange that Lab 0 didn't roll over to this term. Let's go on and talk about Lab Module 1, Ubiquity of Microorganisms. Hopefully you can find this online. The primary learning objectives know that microorganisms are almost everywhere, describe the purpose of culture media, describe colony abundance, diversity, and morphology. We're talking about colony morphology. Define the following terms: inoculation, ubiquity of microorganisms, sterile, isolation, incubation, in vitro, culture media, and there can be a broth culture, an agar culture, a plate, which would be an agar plate, a slant, a deep. Both of these are agar test tubes. What's a fastidious organism, a non-selective media, a ubiquitous organism, and what's a colony? Any questions about any of that? So microorganisms are organisms that are too small to be seen with the naked eye. And these do include viruses and prions, which you could argue they're not organisms because they don't have cells, and we'll simply say that they're acellular. It also includes the helminths, the worms in our studies, even though the adults are not microscopic. However, their infective stages are microscopic. know the term ubiquity of microorganisms refers to the concept that microorganisms are essentially everywhere. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We'll also learn basic lab protocols, including the use of solid media, how to label a culture appropriately, procedures for disinfecting the lab benches, and procedures for washing your hands. And that would be a good one just because of COVID-19, although hopefully all of you know how to wash your hands. But you want to do that to prevent the spread of COVID-19. A culture media, also known as a growth media, is a sterile laboratory preparation that contains a variety of nutrients that are required by growing microorganisms. And we use these in the lab. In this lab, you're not going to actually use a culture media, but you need to know how to use them and how to handle them and what they are and stuff like that. So we're going to hold you accountable even though you're not going to be using a culture media. Culture medias may be liquid. These are usually called broths, and they're commonly used in test tubes. You can add agar or agar to the liquid media, and then we call it a solid media. And these we usually call an agar media. Agar is a gelatinous substance that's extracted from seaweed and is added to thicken a liquid media to make it solid. It is possible to use other materials to do this, but agar is by far and away the material most used to thicken a liquid media and make it solid. You can also use gelatin, and we'll talk a little bit about that, or you could use starch, and that one isn't used much. All right, so you have a video to watch preparing a pour plate, so go ahead and watch that, not now, but when you do the lab. Let's talk a little bit about general purpose medias. These are media that are used for general purposes, generally just growing organisms, and they're non-selective in function. They usually have a complex composition, meaning many molecules are in them and we don't know what they are and don't really care. We're just using a general purpose media to support the growth of organisms, many organisms that are non-fastidious, meaning organisms that do not require special things to grow. And these general purpose media usually use peptones, yeast, or beef extracts to supply the nutrients to the microorganisms. A fastidious organism is one that you have to add special things to the media in order to get a fastidious organism to grow. Like, is it Streptococcus pyogenes? You need to add blood or specific nutrients in blood cells, or else the Streptococcus pyogenes is not going to grow. Any question about that? So Streptococcus pyogenes is a fastidious organism. E. coli is not fastidious. It will grow on most media. There are 3 general purpose media that are commonly used in microbiology. Nutrient agar with yeast extracts added, and that's a little complex to say, so we usually just abbreviate it NA for nutrient agar with yeast extract, or tryptic soya agar, and it's sometimes abbreviated TSA, and then nutrient agar, or the abbreviation NA. And these general purpose media grow most microbes, but not all microbes. They do not grow anything that's fastidious. When you're growing something in the lab, it's it's required, although for you guys it's only, recommended, because you're not really labeling things, but I'll quiz you on how you label something in a microbiology lab. It's vital that Petri plates and tubes are correctly labeled so that we can know whose it is and how old it is and what the media is. And so you need to add your name or your initials, the date that you're using it, the type of media, like NA. The incubation temperature is optional, and then the inoculum, what you're growing. If you know what it is, write down what it is, like E. coli. But if it's something like something you're growing from air or water, write down water or water treatment, something like that. You should label things on a test tube by getting a piece of tape and then writing on the tape. It's good to write around the perimeter so you're not blocking the plate so that you can't read it. The inoculation is the placement or transfer of a microbial specimen into the culture media. You can use various tools for inoculating something. Incubation is the placement of the inoculated culture media in the appropriate physical environment, usually called an incubator, for a specific period of time to allow the microorganisms to grow. And this is an incubator, and we have those in the microbiology lab, and I'm pointing that out because you'll never probably see it, but if you were actually going into the lab, you grow your cultures in an incubator. Incubators can change the oxygen, the carbon dioxide, the temperature, the humidity, and the time. Our incubators are more simple, and they really only control the temperature and the humidity, as well as the time. That's how long you grow it in there. When you grow plates in an incubator, the agar in the plate has moisture in it, and the moisture will come out of the agar and then condense on the lid. I don't know if you can see that. And then will form droplets of moisture on the lid, which will then drop down into your culture. Assuming that the lid is sterile, that won't contaminate or necessarily contaminate the culture, but it will spread your culture and could ruin it. And the other problem is the very edge of the lid may not be sterile, and getting that liquid down near the edge could drop from the edge and then could contaminate your culture. So you don't want the contamination either. And if you have the moisture dripping down, it certainly will increase the risk of contaminating your culture. And like I said, it'll smear all your colonies and make a mess of your culture. So we don't want that. What you can do with a culture, the Petri dish, to prevent that from happening is, instead of growing them, sorry you can't see this, get it up here, instead of growing them, putting them in the plate right side up, put it in the incubator upside down, and then there will be agar on the top. And then when the moisture comes out of the agar, it'll condense on the top where there's agar, and then the agar will wick the moisture throughout the agar, and so there will never be a drop forming that won't drop down. And even if it does drop down, so what? It drops down onto the lid, and that wouldn't be a problem because you don't have any microorganisms growing on the lid. But it won't even drop down because, like I said, the agar will wick the moisture throughout the agar, and so you won't have a drop forming. So we grow all Petri dishes in an incubator upside down. And that's to prevent the moisture from condensing on the lid and then dropping down. Any question about that? Bacteria colonies generally have a smoothly textured surface and a distinct boundary in comparison with fungal colonies, which tend to be furry or powdery or spiky. And so you can usually look at your colonies and determine whether they're bacterial in origin or fungal in origin. And there's a fungal colony and there's a bacterial colony. I should mention that bacteria colonies tend to be formed by one cell or a few cells. And if it's by one cell, that cell will divide into two, two will divide into four, four will divide into eight, etc. until you get at least 1 million cells, maybe hundreds of millions of cells, before you get a colony. So the smallest colony here, probably that one there, has at least 1 million cells. Any question about that? And each of these colonies initially started by one or a few cells. And then that cell divides to form the colony. Any question about any of that? There's a video you can watch on how cells divide by binary fission. That'll be part of the lab. You do need to know about colony characteristics, but I will give you this table, and I don't know what's wrong with this web page here. I noticed this earlier. It's not displaying correctly. And I'll give you this table. You need to know how to use this table to give the colony characteristics. And I won't ask you, is the colony margin, that's the edge of the colony, undulate or lobate? Those are too close together, but I could ask you, is it entire or lobate-like? Okay? You do need to know what abundance is, the total number of colonies in the agar plate, and the diversity, the different kinds of colonies on the plate. Like in this picture here, the abundance of cats is 7. 1, 2, 3, 4, 5, 6, 7. And the diversity is there's 2 species of cats, cheetahs and lions. Know the terms for this lab. You don't need to look at the reference notes. And then find the worksheet and write up your answers for the worksheet. Let me see if I can show you that. I'm not sure if that's it or not. I can't read it. Nope. I'm not in the right place. Well, get the worksheet, answer the questions, and then submit the worksheet by Saturday. And there's the worksheet. And this will be worth, I think it's 2.5 points. All right, any questions? If not, I will be here at least until 7:30 to answer questions. Go ahead and work on Lab 1.